all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So there are two things to follow up on Novavax. There was one that is the myocarditis and the incidence of that comparatively between Novavax, Moderna, Pfizer, and unvaccinated. So I'm working on that. The other one was this preprint that John Snyder said yesterday that Texas Meg had posted in Discord. Texas said John did it. So someone posted this, and I think it's an interesting preprint. I'm going to discuss that preprint from Novavax, talking about their booster and the efficacy of it. Let's look at the preprint first. So I want to start from the funding because many times when I have done a talk about vaccines, folks ask me that what was the funding. So the funding is from South African healthcare. So PLM is supported by the South African research chains, then Bill and Melinda Gates Foundation, and I believe there is Novavax as part of the trial as well. And then they say at the end, the phase 2 clinical trial was funded by Novavax and Bill and Melinda Gates Foundation. The findings and conclusions contained within are those of the authors and do not necessarily reflect positions of the policies of the Bill and Melinda Gates Foundation. At the same time, if you actually look at the references and competing interest, you would see that some of the authors are actually previous employees of Novavax as well, some from Regeneron and so on, some BioNTech and so on. So there are folks who are from the vaccine industry that are in it. So I consider, this is my opinion, this is a Novavax's study about Novavax. So with that, let's look at some of the things that I have. So these are the gifts for humanity, and they're continuing. Here is drbeen.com. This is the one-time fee. It should not be such a low fee, $67 one-time fee. So yes, it's always a issue with my marketing team. They think it's not right. Anyway, so here, Novavax triggers potent neutralization of Omicron sub-lineages. I will add to their title by saying after a booster dose. And I would further say that how long is not known. Then also, this is an in vitro study. So the serum samples or the blood samples were from vaccinated individuals, but the neutralizing titers and the titers are measured in vitro. I was also not very much impressed with the quality of the write-up. Although we have been looking at many preprints, and preprints are usually not the highest quality, they need further work. But within the preprints as well, there are lots of gaps. For example, if you click on the PDF here, you cannot actually go to the PDF because of whatever reason. So somewhere over here, there is a PDF link and you cannot go to that. Similarly, there are some spelling mistakes, grammar mistakes and stuff. So let's leave that and let's now look at what they are talking about. So Novavax, look at my own spelling mistake here. Novavax COVID vaccine and Omicron. So NVX-COV2373 is the vaccine. This is a preprint. Here is a summary of the preprint. Here is what they're saying. They're saying that their vaccine, after two doses, not the booster, after 2 doses of Novavax, the neutralization capacity or capability or ability of the vaccine against BA1 was, and they use the word 72%, that it looks like, well, that was 72%, but actually it was 72% resistant. So it was low. So 21 samples out of 29 samples were resistant to Omicron. And the antibody titers were low, 34 GMT. Again, so the same two dose of Novavax, the serum samples and the antibodies there against BA4 or BA5. That was 59% resistant. So 17 sample out of 29. And the titers for the neutralizing antibodies was 47 GMT. Why did they talk about their own vaccines' low efficiency or performance for these variants? They are trying to make a point. Their point is, with the two doses, we do not cover. So, this is my interpretation of their point. They are not writing it that way. Their point, my interpretation is, with two doses, we do not cover Omicrons very well. However, if you add a third dose, then our Novavax's performance becomes equal to Moderna as well. Not Moderna, Pfizer or messenger RNAs. That's the basic message. So first they present to you that the performance isn't very good with the two boosters, although they say that the severe cases in the trial were protected 100%. So the patients were protected, or vaccinated individuals were protected from severe cases by 100%. So then what is the situation with three doses or with the booster? That is what the study is about. So of course there is samples from individuals with three doses, and the result, the protection, is not seen in production. Instead, it is seen in vitro. So we'll discuss that. But here, if you see, the antibody titers are 1197 GMT against BA1 and 582 against BA4 and 5. And they say, and you would see that a little later, they say this is comparable to mRNA vaccines. So similar in magnitude to 3-dose mRNA vaccines. So yes, this is also important that the vaccine, mRNA vaccine, was also 3 doses. So what is their conclusion of this preprint? Their conclusion is Novavax for booster. So take a booster. That's what they're saying. Also, they're saying this is an inexpensive vaccine, and they're saying that it does not need cold chain. That told me that there is a little bit of a frustration, I think, on the company's part to try to push the vaccine. So they are actually talking about other benefits of the vaccine in addition to the vaccine's current presentation. So this is it. If you wanted to see what is their message, they are saying our booster performs as good as messenger RNA vaccines. But 2 doses do not perform very good. However, they perform well to protect against severe disease. This is it. Done. So, details. So they start their paper by saying that 90% efficacy in the trials and 100% against severe disease, which we know we have discussed that in the past. Now what they did was they took a number of people. They do not give exact number, but they say samples are 29. So I would expect these are 29 people. And they took after the vaccination, they took their plasma samples. Novavax, they took samples 14 days after the second dose and 35 days after the third dose. So about a month after the third dose. In case of BioNTech, they took 2 months after the second dose and 1 to 3 months after the third dose. That is interesting for me. I would have loved to have the actual numbers to be able to compare them, but those numbers are not present and I could not get to the PDF. So with these samples, what did they do? What they did was, then they took the original genetic message of the, or RNA of the Wuhan variant spike. Then they modified that spike to produce the variants or the spikes for the other variants. So just a quick look at it. Here, if you go down to the methods, you would see here the SARS-CoV-2 Wuhan 1 spike cloned into pcDNA 3.1 was mutated using following kits, and it included the following mutations. So again, a few days ago, we were very upset that Boston University performed some mutations, and that was bad. But this kind of mutations, changing the RNA and then combining that RNA with the viruses, it does happen. Yes, there is a huge risk with that in the kind of work that Boston University did. So here, they took the original RNA, they modified that RNA to produce the spike proteins of other variants. Then, instead of actually making the whole virus, then they created pseudo virions, and that is also a um, routine or practice in the labs, that instead of making the original variant, which is going to be really dangerous, so what they do is they make lentiviruses. Lentiviruses are generally innocent viruses, and then they change their DNA to add the spike protein DNA in them. So they add the spike protein plasmids in them, or fuse with them, and so these viruses start exhibiting these spike proteins. Then they also had cells that were overexpressing ACE2, so that these innocent looking viruses can bind with these cells. And of course, and they had the antibodies that were produced by the people who were vaccinated by Novavax or Pfizer vaccines. So then they put those samples in this structure to see how much protection these antibodies are going to offer to prevent this little virus from binding with the cell. Right? So this is the basic method. So it is, the samples are from people, but the rest of the experiment is in lab, it's in vitro. And here is what they had. And this is from, of course, their site. So they did many tests. I just want to focus here. So Omicron BA1, this column here is the 2 doses. This column is 3 doses. So Omicron BA1, 2 doses. This red one over here, the red part shows the amount of resistance, and we have already seen the numbers. So for example, against the Omicron BA1, 2 doses produced only 34 GMT, or low levels of titers for antibodies, and the resistance was high, meaning it didn't perform very well. But if there was third dose, if there was a booster, then you can see that the antibody level, sorry, I should be here, antibody level is 1197, and the protection was good. And if you see here, the pie chart, the resistance was almost zero. They actually do not put the correct number there to say 0 or 5 or 1. They just say, here is a pie chart, you can read it. Similarly, with the BA4 and 5, 2 doses of the Novavax, this is the situation, so not very well, plus there is resistance. Or you can call that escape. And here with the 3 doses, the titers are great and resistance is less, so that is all interesting. So this is what they are saying. And they have a similar behavior for other variants as well, but really Omicron is what they were working with. Then, if you see here, this is a comparison of Novavax with Pfizer and also adenovirus-based vaccines. So I'm going to leave this because I'm not a fan of adenovirus-based vaccine anyways. So let's look at these two. If you see here Novavax first, so BA1 is dark red and BA4 and 5 is orange. So this is BA1, this is BA4 and 5. So Novavax 3 doses, again against BA1, 1197 and 582 against BA4 and 5. So compare that to this side. So if you see here, BioNTech, against BA1, 1078, and Novavax was 1197. And similarly, against BA4 and 5, Pfizer BioNTech was 667 GMT of the titers, and the Novavax was 582. So very comparable. So from this comparison, they are saying that hey, we are producing neutralizing antibodies with the similar titers as the other tried and tested and in production vaccines. Then they add some more data, which is interesting. Again, I think that this is more of a hey, our vaccine is good. Number one, they say that 6 months after the second dose, the binding antibodies were observed. And they then say further that possibly further maturation would continue to occur. So Novavax have seen in their FDA debrief as well, they insist on a affinity maturation, plus they insist on the cytotoxic pathways, the T helper 1 pathway. So here they're saying that, hey, after 6 months as well, second dose, we are still making antibodies that are neutral, that are binding antibodies. So hopefully these contribute towards protection from the severe disease. Then, and they use the word may have, I changed it to hopefully. So then they also say that the CD4 memory cells, so remember T helper cells, so CD4 memory cell and CD8 memory cell were robust. This is their term, robust, in 100% of the patients, CD4, and CD8 memory in 65% of the patients or vaccine receivers. So that was their CD4 memory and cytotoxic T cell. So non-humoral arm, cytotoxic arm. Then they also talk about FC function. So I hope that we know this from our previous discussions, that when we have an antibody has fraction constant, or this part, which is the constant part, and here there is complement activation, then the fraction constant can attach with the macrophages and other things and help, pick up the viruses or bacteria or whatever antigen is present in the environment. So these are called biological functions of an antibody. So they're saying that the FC functions of these antibodies are an additional component as well. So FC will be true for every vaccine that produces antibodies. So nothing special to them, but they use it, they invoke it. And they say, you know what? Because of the FC function plus neutralizing antibodies likely limit this work, this behavior will likely limit viral replication to prevent severe disease after breakthrough infection with Omicron lineage. However, I want to read the statement to you which they wrote, because there is a grammatical mistake, and so it's kind of, you'll have to figure out how you want to read it. Here, they say, this T cell and FC effector function data, coupled with the high titer neutralizing antibodies we have described here in this study, suggests that this vaccine, then they use the word will, is likely limited. So future, present, and a third term here as well. So I think they wanted to write, suggest that this vaccine will likely limit viral replication to prevent severe SARS-CoV-2 disease, breakthrough infection with currently dominant Omicron lineage. But they have a little mess up over here. So this is the discussion. If you said, once again, what is the message? The message is, the summary is that this is an in vitro study where they have taken the plasma from people vaccinated by Novavax, 2 doses and 3 doses, and the Pfizer, and even the adenovirus based vaccines. And what they're showing is that 2 doses against Omicron are not very performant. I don't want to say efficacious because their trial shows efficacy. So I'm just going to use, just like they're using robust and other terms, I'm just going to use performant. However, their third dose brings them equal to mRNA. The question that is still out for me was how long? Is it 2 months long? Is it 3 months long? Is it 16 weeks? Is it 90 days? That is not known because this was an in vitro study. So that is the discussion. Tomorrow I have a meeting, so I'll do a lecture earlier in the day, or I'll skip the lecture and see you guys on Monday, all the cool beans. Please like, subscribe, and share. There are links in the description if you would like to buy Dr. Bean's access for a very, very low price. Or if you would like to support this work, you can buy me a coffee, you can use PayPal, you can become a member of Substack, you can even become a member of this YouTube as well channel. So let's see some questions and then we stop. Daniel says, can endothelial dysfunction be considered as long COVID? Long COVID has a component of endothelial dysfunction, actually a major component. However, everybody's pathology is different, but endothelial dysfunction is a participant in this in majority of the pathologies that I have read so far. Bob says, so can IUN vax get Novavax or not? So did you mean mRNA vax? So they are a booster for any other vaccine and for Novavax, if that was the question, Bob. Oh, sorry, unvaxxed. So yes, so unvaccinated can get the Novavax. I do not know can unvaccinated get Novavax or not? Yes, so it is a vaccine as well. The difference from the messenger RNA type vaccines is it doesn't have any messenger RNA. Plus, somebody asked me this question, and the question was, are you really sure that there is no fetal cell involved? So there are no fetal cells involved because this is actually made in moth cells, which people actually made fun that will I become a butterfly? Moth cells, so no fetal cells, plus the vaccine has spike proteins themselves, but not messenger RNA to make our body cells make spike proteins. So that is the vaccine structure. Let's see some more questions. Julian says, greetings back to you. So Nipa says, by now most are vaccinated or infected. Who have they worked on? So this is an in vitro study. There are folks who are still not vaccinated and still not infected, or at least they do not know that they are not infected. Although, Nipa, to your point, the seroprevalence has been above 90% in many areas. That is correct. There are many. I actually read a comment yesterday about someone who said, I'm still not vaccinated, but I will have to get vaccinated because of a mandate. And they said, it is good to understand more about the Novavax. So with this, thank you very. Okay, so Cynthia says walk. Yes. So we will have our walk on Saturday. I have a meeting at lunchtime tomorrow and then one in the evening. But Saturday morning I'm free. Saturday evening it is actually funny. I'm going to drive with my son to San Francisco. So very good. So hope you liked it. Like, subscribe, and share. And the links to support in the description. And I would see you either earlier tomorrow or on Monday. Bye-bye for now.